Let me first of all welcome you and introduce our panel. So from my right to left, we have Seyoum Ayehunie and Seyoum is a tissue engineer. He's made many 3D models for toxicology studies, and he is from a company called MatTek in the Boston area. Next to him, we have Uwe Marx from Berlin, company called TissUse, which is a nice little marketing trick. He's a physician and very much involved in technology development for these advanced cellular models. Then next to him is Mayo, senior scientist at Sartorius, also in the Boston area, cell biology and technology enabler, I think she would describe herself as, so helping the workflow to get these advanced tissue models into the systems. And finally, we have Zaher Nahle, who's the scientific advisor, I would say. He runs a company called Ivyctory, which is also quite nice. Originally a biophysicist, now a science policy expert, has thought a lot about FDA regulations and so on. So, to set the scene, we're thinking about how we shouldn't be using so many animals in research, in drug development, in toxicology studies, and how we can do this, the technologies in biology that now allow us to make better alternatives, not just animal alternatives, but actually scientifically better alternatives to the animal studies that we've been using for such a long time. And Martha will be kicking things off by explaining to us exactly what these new technologies are in biology. But thinking about the challenge, I think in 2022, that's the latest figures that we have for statistics from the European Union, there was approximately 7 million animals used for scientific purposes. And 70% approximately of these animals were used for basic and applied research. 50-50 basic and applied research. And I would say that drug development is a big part of that applied research. And then 13% are used for regulatory purposes. That is to bring your entity onto the market. You have to overcome regulatory hurdles for safety, quite rightly, etc. Sometimes they can be problematic, and we'll hear about that at the end. So my name is Alison Abbott. I'm a science journalist, and I'm based in Munich. And now I'd like to learn a little bit about you so that we can pitch things properly before we start. So can you please put your hands up if you are a biologist? Okay, so put your hands up if you are not a biologist. Okay, good. So I think we have something for everybody. We can talk a little bit about the biology and the challenges, but also we're going to talk very much about the context, because at the end of the day, we want healthy societies, and that's the direction that we're going in. So let's start, please, at the beginning. So Martha can tell us what on earth we're talking about when we talk about advanced cell models and organoids. Just a bit of technology, Martha, please.
Sure. I think what we're seeing now is kind of a convergence of multiple disciplines that have started to really take off in parallel. Stem cell engineering, tissue engineering, synthetic biology. So all these kind of are converging and helping enable these advanced cell models. You may have heard of organoids, microtissues, things like this that are better at informing on human biology. They better represent the genetic and ethnic diversity that we all represent, and really help inform on the physiology and human response to therapeutics and so on.
And can we maybe go a little bit deeper into how they represent better our world, our diverse world? I mean, how physically can you do this with developing stem cells?
So I guess in the clinic you can take a patient-derived system. There are patient-derived organoids. Of course, that would represent some of the tumor microenvironment as well as some of the background of the human genetic, and that would change tumor to tumor, person to person. And so that's just one application of these things, and you can therefore screen for personalized treatment, personalized medicine that will best affect the tumor, for the individual.
So is it that you would take a sample from a patient which has a unique DNA, a unique epigenome, and then do something magic to make a preparation that can be popped onto some sort of structure?
Sure.
How does it work?
Yeah, that's one application. Those are kind of the tissue-derived kind of systems. There's also iPSC-derived systems where you could take a sample of a blood sample, program that into an iPSC cells that will have the background of the individual, and then generate organs from that. So truly kind of able to take the diversity of everyone into account.
Right. So we've heard a lot about these pluripotent cells in the last X years, 2007, when Dolly was first cloned. So I know that there's different sorts of sources of making these models. You talk about iPS cells. These are the cells that you sort of program them back to an early stage and then push them into the sort of organ you want to develop, like a liver or whatever. But are people still using embryonic stem cells, which of course do have a lot of advantages? So what's the situation there?
Yeah, I'm seeing. I'd say I'd see mostly iPSC-derived kind of systems. I do see some embryonic systems as well.
Mm-hmm. And so, Uwe, tell us what they look like, because I know in his pocket he actually has something to show you.
Yes, okay. We also are using iPS-derived cells. Also, a number of people are using embryonic cells, and I think the regulation with embryonic cells is very different in some of the countries in Europe. So what we are doing, we are kind of combining different organ models or organoid models on this kind of small chip-like backgrounds. We have a small heart here, and then you can start to microfluidically perfuse the organs, and that allows you to run really vascularized organs on chips. And we think that this is with 1, 2, 3, and 4 organs we can do that. Our main goal is to make a body on a chip, because if some of you donate blood and use the iPS cells to make 10 of your organs, we can bring your individual organ on hundreds of these chips. Our downscale factor for a human body is 100,000. So the bodies on the chips, or the organs on the chip, are 100,000-fold smaller than yourself, and therefore need only feeding of what you 100,000-fold of you need. And that is the vision in the future. These 1, 2, 3, and 4 organ chips are running already in industry and in research laboratories, and we will talk about the hurdles for regulatory application.
Right. So are these chips, they're mostly used for drug development, or?
We use it, our customers and research laboratories, primarily in Europe, the US, Brazil, and Asia, they use it for both basic research for new discoveries. This is kind of Nobel Prize winning stuff, because here you can reach to the brain and see it not only from the blood side, but also from the interior of the brain side. We have a cosmetics industry already since 2009 because of the ban of animal testing in cosmetic industries with us, which tried to validate it for liver-skin combination. And primarily we have pharma industry, which are interested to use it for efficacy and safety testing.
Right, right. And so what's the limit of that? I mean, you talk about you get a whole body of every individual in the world on a chip eventually. Presumably this is music for the future.
The whole body is music on the future, but the concept is that each and every organ in your body is built by smallest functional units, and each of the functional units is representing the organ. That's why with 10 liver lobuli, for example, we can fully model the liver. And if you make an organism, like an individual of each of you, you then have everything in place. The limits in our view, the long-term limits, with this kind of organoids, body on a chip, is the size. Some of the things we are not able and are not even targeting to model. We don't want to have consciousness and empathy in the chip. And therefore you need a 1.2 or 1.4 kilogram brain. The brains are too small to make that. So the limits are the size, the same as for heart beating. 300 gram of heart, you cannot build 300 gram of heart beating in the chips, and the same as broken bones, larger kind of pressure-based system. We can do pressure on bones, we can make bone and bone marrow on the chips, but we cannot mimic a large bone breakage, for example. That are the limits, nothing else.
Really nothing else? I would have thought that maybe...
The regulators? Okay.
No, not the regulators. We're still on the biology. But when you have a body, what the kidney does affects what the liver does and what the brain does.
Yeah.
And can you actually reproduce this on a chip?
Yes. Yeah. Our most advanced, worldwide most advanced chips is an PBPK compliant 4-organ chip, which has an arrangement of the human intestine, the liver, and the kidney in the ADME cascade in order to make ADME. Background, and we added, this was questioned by the pharma industry, a brain, because they wanted to have the blood-brain barrier and the brain. For some of the most recent, most impressive toxicities are secondary neuronal toxicity. So the primary drug going through the liver is transformed into a secondary metabolite, and these are toxic for the brain. That's why this chip has a brain in addition to the ADME cascade.
That's really cool. That's really cool. A limited brain, I would say.
It is still a limited brain, and we don't have innervation. We don't have yet blood production hematopoiesis. We have a bone marrow on a chip, which is producing hematopoiesis over weeks and months, but we didn't interconnect it into a closed vasculature for the other organs yet.
Okay. So, right. Okay. And Seyoum, you have also an example of something that you've developed on a chip. Do you want to explain that?
Yes. So these advanced tissue culture models, they are not uniform right now. We have 4 different models. One is spheroid, which is a collection of cells, and the second one is organoids, which have a growth pattern inwards. The differentiation is inwards, it's not outwards. So you cannot do direct application of drugs. And the third one is organotypic type of tissue models, which we develop. They have both the apical and basolateral surface, so you can add drugs topically, or you can add drugs from the basolateral side to mimic systemic delivery of drugs. And the fourth one is organ on a chip. So the organ on a chip, when it was developed first, basically the concept of the organ on a chip was, DARPA came up with a concept that, say, the current models that we have are going to have, it takes 12 years to develop a drug, and it also is expensive. You have to spend about $2.5 billion to get a single drug. So if it takes 12 years, the current animal testing models, which takes about 6 years to get into the clinical trial, they said that doesn't give us time if there is an emergency. So for emergency purpose, if we have this organ on a chip type of system where different organs are connected into one type of chip, then we can address at least very fast. We can see the toxicity and the efficacy of drugs. That's where the concept came in. And so you can now in a microfluidic system, you can put these organs together. So we put some of the organs that we developed at that time were with Uwe, with the company. We were just putting those tissues inside these chips. And we were putting liver, kidney, skin, and intestinal tissue. Different types of skin. Yeah. Like full and skin. It's an intestinal tissue. So most of the drugs, 60 to 80% of the drugs are orally administered. So the intestine is the one which is going to be first exposed for the drug. So it has to be absorbed by the intestine and then get to the liver. And from the liver, it goes to the target organs. That's the whole concept, so that you can see the whole effect within these small chips. Mm-hmm. So that is what the value of these ones are. So the advantage of these advanced cell systems is that the cells are human origin. So they are mostly, in our case, we only use adult cells that are taken from humans. So they have the physiology of the human body. So they respond similar to the human body. That's one thing. And two, you can put these cells, you can put the cells, expand them large quantities, so that from one single donor you can make thousands of tissues, or so you can test different type of drugs. So you can put it into high throughput format. And these models, we think that they are more predictive of human responses, and we have shown that for drugs that has passed for as safe from based on the animal clinical studies, and then when they go to the clinical trials, they failed for the pharmaceutical industry. So we got those drugs from the pharma and test them on these models. So it's a human-to-human comparison. It's not a human tissue model to animal comparison. That's the difference. So we were able to predict those drugs correctly, that they were going to be toxic, or somehow they will have some kind of problem in the humans. So those are type of answers that you will get. In animals, there are two problems. One, they pass some of the safe drugs as unsafe, or you have unsafe drugs passed as safe drug, which the pharma industry has to recall later on. And that is extremely expensive. I can give you one example. The Vioxx drugs that was developed by Merck. It's an arthritis drug. It has been given to people, and then it was pulled out because of heart attack problems. After 1,400 people were died, and Merck was there was a lawsuit, and they have settled it with $8.5 billion. So those are type of things that you can early anticipate not to happen in the human, and you get good responses, and you can model, you can design it early on. You can improve those drugs.
So I think remembering back to my studies and also reading the newspapers, I think that drugs that show toxicity in humans are withdrawn by the FDA, even though for maybe 85% of people that get it get a huge benefit with no toxicity. And being able to sort these people out so that you can so you don't have to withdraw because 2% of people have a bad effect, but you can actually, I guess that's also personalized medicine, isn't it?
That's it.
And so would your chips, your organoids, help you anticipate who your treatment population is and who should be excluded? Or is that too difficult from the regulatory point of view?
I think for those type of assays, you have to bring it to personalized type of medicine. For example, now we are working on tumors. We are taking direct tumors from patients. And then we are going to, the first thing that you have to do is you take this tiny tumor, which is about 4 millimeter, and then you have to expand it in culture 1 million times. So you have to create that condition. So we are able to expand from colon cancer. We are able to expand now large number of tumors. So now what that will give you is, for that part, it's not like directly to personalized medicine, but this is a development phase. So what that will give you is ultimately is if we have that type of, for example, breast cancer has 119 drugs, and they have been tested in humans, so you don't have to go to human clinical trial for those type of drugs. These are repurposed drugs. So you can pull all 119 drugs for breast cancer, and if we have these tumor models available, then you can test everything at the same time. So that, for example, for metastatic cancer, the physicians, they may have first line, second line of drugs. After that, it's anybody's choice. So you can screen everything, and you can tell the patient, the doctor, you can advise the physician which drugs are going to be good for this particular patient.
Yeah, so it is going in this direction. So the other thing about using animals is that you need large numbers of animals to get your statistics. And I'm wondering, Martha, maybe you could explain a little bit about this. How does the robustness of one particular type of stem cell represent what animals used to represent in large numbers? That is, if you take a sample and make a vat of this particular cell type, every time you did that experiment, would you always get the same results because it came from the same pot? Or tell us a little bit about the robustness.
Yeah, I think that is one of the hurdles. The workflows can be quite cumbersome, expensive, and there is some variability at times. And to your question about the donors and the toxicity, I think this is kind of a place for these models to shine, but there needs to be some regulatory guidance, at least with the FDA, right, as to what's a good cross-section of donor representation to catch the toxicity profile. That may differ between genetic groups, ethnic groups, but the workflow themselves do need some polishing, to say, and as tech enablers, that's something we work closely with collaborators to try to identify what they're trying to do, iron out these issues, and move forward.
So you mentioned the regulatory authorities. Before we come to audience questions, I want to talk a little bit about changes there and changes that have been made and not implemented and changes that are still needed. So Zaher, maybe you could start off that.
Yes, thank you. And I would like to say that it's wonderful that the Falling Walls is highlighting this topic, and kudos to Sartorius for actually proposing this particular subject. I boarded a plane, Alison, to come here, and had someone told me that there is a 95% chance that my flight will not make it, I would not have taken that flight. But that's exactly the model we still operate in preclinical drug development. 90 to 95% of drugs fail in clinical trials once they move from animal studies, although they have perfect safety and efficacy profiles in animals. That's really the root cause of the problem. And to talk about regulation, let me start from year 2021, because that was a formidable year for NAMs, what we refer to as new approach methodologies. Some people call them non-animal methods. Regardless, these alternatives in 2021, something wonderful happened in Europe. In the European Union, for example, enacted regulation, resolution B9-0425, which was a action plan and also roadmap for all member states to start deviating from testing on animals that is inefficient, and that plan contained education, training, stakeholder engagement, and also enforcement. So that was wonderful. 6 months before that, in the United States, the FDA Modernization Act was introduced in March in the House. And that was also a historic draft bill that replaced the word animal testing with non-clinical testings, and legislators went on to define what that means so that there's clarity about this issue, and that is organ chip, AI, 3D tissue engineering.
Right, so if I've understood that correctly, you're saying that this new legislation said it is no longer mandatory, which it used to be, to use animal models.
Correct.
Yeah.
Correct.
Okay.
It's a simple change to the 1938 FDCA. Imagine this, our laws are from the 1938, which is the Food, Drug, and Cosmetic Act of the United States. So that was a major development. It was later introduced in the Senate. As you know, in the United States, both chambers have to approve a legislation so that it becomes a law, and it was signed into law in December 2022. Typically, a legislation like this takes 7 years This was done in a year and a half, and that is a testament to the organizations that were pushing it. And in the interest of full disclosure, I'm a scientific advisor for the two organizations that were behind this, the Center for Humane Economy and Animal Wellness Action. So, kudos to them for their resilience and their ability to assemble a bipartisan group, really led by Senator Booker and Senator Rand Paul in the United States. So that became law, Alison. But then we have a problem. Now we are approaching the second year anniversary, and still the regulatory authority, the agency, has not initiated rulemaking. Rulemaking is a process that takes the law that's enacted by the legislator and transforming it into language, guidance, instructions, so that we have regulatory clarity. So that when drug developers want to go and propose their NAMs systems, they can do that with confidence and with the full authority of the law. So it's really indefensible that this has not happened, which led a group of senators to use a local terminology, pissed off, and fired a letter to the agency demanding an explanation. And they also introduced this year what's known as the FDA Modernization Act 3.0, which is a new draft legislation in its full form in the House to enforce FDA 2.0. You see how inefficient that is, because if every law needs a law to enforce, we lose half of our lives, really, in that process. But also to establish a standalone, credible qualification programs for these type of things within the drug development tools at the FDA. And as you know, drug development tools, whether they are methods, measures, or materials, these are tools. They assist the regulators in understanding safety and efficacy of a particular development. Right now, the drug development tools within that agency are biomarkers, animal models, and clinical assessment tools. So the qualification program will create the missing tooth, which is specifically for that and not shuffled into a pilot program that takes one or two. And that will democratize really the process of science and let all of these folks compete, not selectively, but on an even keel in advancing this operation. So this is a quick rundown of the major legislations that are happening now. We have a new administration in the United States. This may change. Now it's too late to do the 3.0, but it will be picked up next year. And finally, I close on this. 2 weeks ago, a report came from the subcommittee on NAMs to the Scientific Advisory Board of the FDA. This is a major report. I have it here if colleagues want to see it. Also, it's available online. And that gave 6 recommendations to the FDA. They are non-binding, but these are their own people, telling them exactly that the recommendations should be the ones that are in the FDA Modernization Act 3.0, meaning establishing a framework for qualification, an office at the FDA that is really tractable and take care of these particular issues, as well as regulatory clarity, eliminating confusion, and also transparency and a database for all the NAMs to be under one roof. So I'm very energized and optimistic that once different entities are saying the same things, then we can move forward to achieve this major implementation of the law.
Yeah, let's hope your optimism is justified. I know that when I speak to people as a journalist, I work mostly in neuroscience, but also cell biology, and in the translation process, people are always talking about the interaction with the FDA, and I did have a feeling that for biologicals, there's an informal system running. Uwe, do you want to comment on that?
Yeah, we are trying to run these systems all the time. By the way, we have a community of microphysiological microfluidic systems running around, and we here in Berlin have the privilege every 4 years to get all the stakeholders here to Berlin with the FDA, with all other regulators, with MIT and all the players. And every 4 years we are publishing our reports. So the next report is already submitted. We need to rework it a little bit, and then you can see where we are, where we stand with pushing these things into regulatory acceptance. And here the important thing is that in this report, this year report, which stems from a workshop meeting last year on the Second World Congress of Microphysiological Systems here in Berlin, we clearly identified that the new biological drugs and therapies, cell therapies, CAR-T therapies, antibodies, they all turn to be human. So they are biological by nature and they are human by nature. And you cannot test CAR-T cells from a patient in animals for toxicity, because animals will react to the human cells. So these kind of biological drugs are really currently our drivers, because no animal tests are there. But the assays themselves are universal. If they work for these cells, they also work for any low molecular weight drug and can replace the current legislation of animal use in this area.
Right.
That is an important part here.
Yeah, yeah. So it seems like such an obvious thing. We put a lot of money into, also taxpayers' money into the development of stem cell biology, and everything is coming together, as Martha said. So what is it about the regulatory authorities that is sort of blocking? Why aren't they welcoming this with open arms? Why are they so nervous?
It's a tough one. I don't know who wants to take it.
Okay.
But I mean, first
Just
of all,
briefly.
it's very important to recognize that these regulators have a lot of responsibilities. They are entrusted with safety and efficacy.
Yeah.
So the system by itself is is refractory by nature. It doesn't accept change that easily because the stakes are high. However, that should not be an excuse for inaction. It should not be an excuse for not being on top of the most innovative technologies. And I refer you to an example that we all can understand. During the COVID-19 pandemic, the International Association of Coalition for Medicine Regulators suspended or allowed the testing in human before animal models can. So when the stakes are high, that was in May 2020, we can go directly to human if we have sufficient data. But when the stakes are not high, then we reverse back to animals. And that's the reason why we had the vaccine for COVID-19 in that very short period of time and not in 14 years, right? So
Is that door still open? I mean, having opened it once, does it stay open?
Well, that's the hope. That was the hope, right? But there is a backtracking on a lot of things, going back to what we know how to do, right? Which is animal models. So clearly, this is an issue that needs to be addressed with the regulators in terms of their speed of incorporating these type of technology. There's an economic benefit that we can talk about if you'd like for these type of chips, and I'll give you an example from 2023, 2021, and 2022. There is also a national competitiveness angle to this, to be on top of technology and advancing.
Yeah.
But there's most importantly a relief for patients who are, some of them are waiting for 15 years. Rare drugs, for example, are 7,500. 95% of them do not have an FDA-approved drug. And with this type of technology, we can accelerate time to market from target discovery and cut it really in half, if not more. So there are lots of benefits when we are talking about making the regulators balance what they know how to do very
Benefit
well with
risks.
new technologies that are emerging. And actually the expectation is that they will be on top of these things, and these laws like the FDA Modernization Act and others should be embraced very much by these agencies, because they have a lot of responsibilities on them.
Now I've got one last question which I have to run past everybody, and that is, let me pull it up so I can say it correctly. What are the, sorry, I'm not being very efficient here. Oh, looked on the original.
Things to change.
Sorry. So what is the next step to break down the wall to fully realizing organoids and microtissues reliable alternatives to animal testing?
Three things: regulatory clarity, investment and spending, and deviation from a reflexive memory we have about animal models.
Very good. Martha?
efficient workflows, problem solving at the technical level that help end users get the amount they want, the scale they want, the physiology they want, and so on.
Uwe?
Getting one after another pharma sponsors and FDA and EMEA into validation, voluntary validation programs.
So?
Early involvement of the regulatory bodies to give us guidance in design, validation, and application of assay systems.
Okay, very good. So it remains for me to thank Sartorius very, very much for sponsoring all of this and for choosing a theme that is really, really important at the moment and has many, many interesting aspects. And I can only apologize for having to keep it short, but otherwise they would have pulled my fingernails out.
Thank you, Alison.
Thank you.